Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of December 2023 shift 2 or evening shift. I will be discussing cell biology, biochemistry, applied biology, methodology, DNA replication, protein synthesis, genetics, etc. in this section. This is part B of the paper. Coming to the first question. The base composition of a genome of a newly identified virus is given below. Adenine 25%, cytosine 35%, guanine 30%, thymidine 10%. Based on this information, the genome of the virus is. Since thymidine is present, it is not RNA. So 3 and 4 can be not taken into consideration. But if you see adenine, it is not say having the same number, same percentage as that of thymine. Cytosine is not the same as is not having the same percentage as guanine. So it should be a single-stranded DNA, not a double-stranded DNA. So that is, and it doesn't follow Chargaff's rule also. So it should be a single-stranded DNA. Then next one. The amount of energy required to break a single covalent bond is. So here both 1 and 2 are correct. 200 to 500 kilojoule per mole and 80 to 150 kilojoule per mole. Then next one. Which one of the following option represent parts of a tRNA that are involved in amino acid charging and interaction with the mRNA respectively? So for interaction with mRNA, we all know it is the anticodon loop. So first and second options we will not take into consideration. But if you see the next one, that is part of the tRNA that is involved in amino acid charging, it is the 3' end. So answer here is option 4. Then next one. An enzyme X converts L amino acid to a racemic mixture of D and L forms. Which one of the following coenzyme is utilized by the enzyme X for this conversion? So amino acid racemase is the one that catalyzes the interconversion of D and L enantiomers of an amino acid. And this amino acid racemase, it requires pyridoxal phosphate as its coenzyme. So the answer here is option 1. Then next one. Which of the following statement about human transposons is incorrect? Lines are autonomous active transposable elements in humans. That is correct. Signs and lines are retrotransposons. That is also correct. Human genome contains many more copies of retrotransposons than DNA transposons. That is also correct. Lines and signs contain LTR, that is long terminal repeat, that initiate transcription. No, lines and signs do not contain LTR elements. So the answer they have asked which is incorrect statement. So answer is option 4. Then next one. Mammalian cells have just crossed the restriction point of the cell cycle are likely to have. So G1 checkpoint is the restriction point where the cell is committed to entering the S phase, that is over here. So RB will be phosphorylated at this state. The cell is committed to entering the S phase, so RB will be phosphorylated. So one high levels of unphosphorylated retinoblastoma protein, that is incorrect statement. An active anaphase promoting complex, that is seen at the anaphase, not at the S phase. Low level of mitotic cyclin B, that is correct. If you see the diagram here, you can see that at mitosis only cyclin B is increasing, and in late phase only the production of cyclin S phase, late S phase production of cyclin B starts. So it is not at the S phase. So low level of mitotic cyclin B is correct. Condensed chromosome are characteristic of metaphase. So the answer here is low level of mitotic cyclin. Answer is option 3. Then next one. Which one of the following statements about ATP generating mitochondria is true? The outer membrane is permeable to protons. That is correct. The inner membrane is not permeable to protons. That is also correct. The intermembrane space has a high concentration of protons. Yes. The matrix has the highest concentration of protons. No. So if you see, the question should have been which of the following is not true. Now since they have asked true, 1, 2 and 3 are correct. Then next one. Which of the following statements about biological membranes is incorrect? Lipid molecules can diffuse freely in the plane of a bilayer. That is correct. The ratio of the size of head group of fatty acyl chain of phospholipids contribute to the curvature properties of lipid membranes. That is also correct. Individual lipid molecules rapidly rotate along their long axis. That is also correct. Phospholipids freely diffuse from one leaflet of bilayer to another. That is incorrect, and your answer is option 4. Then next one. Which of the following exhibits ATPase and helicase activities for promoter opening and clearance? It is TF2H. The tri-SNP particle is composed of. So SNPs are small nuclear ribonucleoproteins and they are involved in splicing of pre-mRNA in eukaryotic cells, and the splicing, it removes the introns and joins the exons. So tri-SNP particle, it is composed of U4, U6, U5 SNPs. Answer is option 4. Then next one. Which one of the following statements about clamp loader is not correct? The clamp loader is a 5 subunit heteropentamer that helps in DNA replication. That is correct. Clamp loading is an ATP dependent process. Yes, the clamp loader, it will undergo conformational change. When bound to ATP, then it will open up and it will load the clamp onto the DNA. In the absence of ATP, this function will not be performed by the clamp loader. Now next statement. In the absence of ATP, clamp loader retains a spiral shape to bind and open the clamp. In the absence of ATP, it doesn't open the clamp. Only in the presence of ATP, the clamp is open. So third is your answer. They have asked which is not correct. The clamp loader and DNA polymerase compete for the same C-terminal phase of the clamp. That is also correct. So they have asked not correct here. So answer is option 3. Then next one. Which of the following is co-translational amino acid modification rather than post-translational modification? Phosphate added to serine phosphatase. So it is a post-translational modification. Selenium added to cysteine. Selenium added to cysteine is a co-translational modification. Answer is option 2. Acetyl added to lysine again is a post-translational modification. Methyl added to arginine is also a post-translational modification. So the answer here is option 2. Then next one. Which one of the following statements about anthrax is correct? This is an acute infectious disease caused by a gram-negative bacterium. It is not gram-negative, it is gram-positive. It is Bacillus anthracis. The beta B subunit of anthrax toxin is known as the lethal factor. No. The A subunit is known as the lethal factor and the B subunit is called as the edema factor. there are 3 subunits for anthrax toxins. Then next one. The A subunit of the lethal toxin is a protease that cleaves the members of MAP kinase family. That is correct. The lethal toxin and edema toxin consist of identical A subunits. That is also not correct. So they have asked the correct statement here. So the answer is option 3. Then next one. Many signal transduction pathways work as molecular switches. On receiving a signal, they switch from an inactive form to an active form. Yes, if you remember GPCR, you can see that the heterotrimeric G protein, the alpha subunit is bound to GDP in the inactive form, and when bound to the GTP, it gets converted to active form. Similarly, there are some other molecules which when phosphorylated they become active form, and unphosphorylated state they become inactive form. Okay. They return to the inactive form when another signal switches them off. Which of the following processes does not directly involve a molecular switch? Proteasome degradation of H1F1A during normoxia. That itself is the answer because it is proteome degradation. The protein itself is degraded. Now nothing is left for switching it on and off. So answer is option 1. Regulation of RAS during cell proliferation. So RAS when bound to GTP it is active form, and when bound to GDP it is inactive form. Regulation of AKT in response to growth signal. So AKT will be phosphorylated in its active form and dephosphorylated in its inactive form. Growth cone collapse regulation by rho A. So rho also it is active when GTP is bound and inactive when GDP is bound. Then next one. Which one of the following statement about different cellular junction is incorrect? Gap junctions are channel forming junctions linking the cytoplasm of adjacent cells. That is correct. Tight junction, desmosome and gap junction together form the junctional complex. That is not correct. That is your answer. They have asked for the incorrect statement. So tight junction, desmosome, gap junction together form the junctional complex is not true. Junctional complex is formed of tight junctions, zonula adherents and desmosome. Gap junctions are not involved there. So the answer here is option 2. Hemidesmosome helps in anchoring intermediate filaments with extracellular matrix. That's also correct. Adherence junction help in connecting actin filaments of adjacent cells. That is also correct. Then next one. Mutants and transgenic models have played a pivotal role in understanding development. Which one of the following statement is true? A transgenic DNA chimera experiment involves the transplantation of cells from a genetically modified organism to another embryo. Only then you can get a chimera. So first statement itself is correct and that is your answer. They have asked which of the statement is true. Then next one. Which one of the following hormone elicits its cellular response by producing CAMP as the second messenger? It is calcitonin, because the principal mechanism of action of calcitonin is that it has got the ability to signal through two transduction pathways once it is bound to the receptor. So one of the pathways it is coupled with CAMP signal transduction. So the answer here is calcitonin. Then next one. Which one of the following amino acid is present in vasopressin at position 3? It is phenylalanine, and vasopressin it is an antidiuretic hormone. So this is the structure of vasopressin. So you can see phenylalanine at position 3. This 1 and 6th position cysteine residues are joined by a disulfide bridge. These are the rest of the amino acids. So it is the amino acid at position 3 is phenylalanine. Then, while developing genetic maps, Alfred Sturtevant proposed that genetic distances are additive. Yes, of course. From test crosses involving two genes, if the genetic distance between genes A and B was observed to be 15 centimorgan, A and oh, I wrote it the other way around. I am extremely sorry for that. A and B it is 15 centimorgan and B and C it is 10 centimorgan. I wrote it in the by mistake. Then the percentage of recombinants observed between A and C would be 25 because it is additive. These map distances are or the genetic distances are additive, so you will get 25 percentage of recombinants between A and C. Given that the arrangement is ABC, ABC is the arrangement. This will be observed only when there is. So you will get a percentage of additive nature of genetic distances only when there is complete interference. So what is interference? If there is a crossover at one spot of the chromosome, this decreases the likelihood of a crossover at a nearby spot. That is called as interference. Now, complete interference means double crossovers will not be observed. Only single crossovers will be observed. So, if double crossover happens, then additive nature of the genetic distances will not be observed. So instead of 25, you will get some other value. Since they have asked, this will be observed only when this additive nature will be observed only when there is complete interference. That is when double crossovers are not there. Then next one. Which of the following statements about determination of ABO blood type is incorrect? ABO blood types are determined by 3 alleles of one gene. Yes, that is correct. 4 blood group phenotypes are produced by 6 genotypes. 4 blood group phenotypes, that is A, B, AB and O. They are produced by 6 genotypes. Yes. Parents with type A and type B could produce an O child. If you see the diagram, the photo over here, parent AO, BO, if you cross, you will get a child with O phenotype. Then universal donors with O blood type do not carry antibodies against A and B. Universal donor like O, it doesn't have antigens but it has antibodies. So the statement 4 is incorrect. And they have asked incorrect statement. So answer is option 4. Then next one. DNA from a strain of bacteria with genotype A plus B plus C plus D plus E plus was isolated and used to transform a strain of bacteria that was A minus, B minus, C minus, D minus, E minus. The transformants were tested for the presence of donated genes. The following genes were co-transformed. Which one of the following option gives the correct order of genes on the bacterial chromosome? Now if you see A and C, they transduce together. So A and C will be written first. So 3 and 4 need not be taken into consideration. We will see 1 and 2 options. So A and C first. Then with C, it is B that is going. So first option also went wrong. It is A, C, D they have written, but actually it is A, C, B. Then with B, you can see D is going. So with B, D is going. Then with D, E is going. So D, E. So answer here is A, C, B, D, E. Answer is option 2. Then next one. Monozygotic and dizygotic twins are used to study the onset of mental illness. The influence of genetic factors on these diseases can be done by calculating the concordance rate. Concordance rate is like the chance of offspring having a certain disease if the mother is having it, or if the father is having it, or his or her elder brother or sister is having it. All these can be calculated using concordance rate. Answer is option 2. Then next one. Which of the following statements about the molecular hypothesis, clock hypothesis as proposed by Zuckerland and Pauling in 1962 is correct? So the concept of molecular clock, it is based on the hypothesis that the DNA and protein sequence evolve at a rate that is relatively constant over time and among different organisms. Now read the statements. DNA and protein sequences evolve at a relatively constant rate. That itself is correct. So they have asked for the correct statement. So answer is option 1. Then next one. To evaluate insulin-dependent diabetes, clinicians prefer evaluating monoglycated hemoglobin referred to as HbA1c, which occurs due to non-enzymatic reaction between glucose and a few amino acids in hemoglobin. So glucose is bound to some amino acids in the hemoglobin. Now which are the amino acids to which hemoglobin is bound? You have to answer that question. So it is valine and lysine that are involved. Answer is option 4. Which one of the following strategies will generate the most precise mutation at the predetermined location of a plant genome? So most precise, that is CRISPR-Cas9 editing will help. tDNA insertion, transposon mutagenesis, all these will result in random insertion only, not precise. So the answer here is option 1. Then next one. Using patch clamp technique, one can find the ligands that could influence specific ion channels. How many molecules of acetylcholine are required to open an acetylcholine receptor ion channel in such an experiment? So how many molecules of acetylcholine will be required to open acetylcholine receptor ion channel is 2 acetylcholine molecules. Answer is option 2. Then next one. Which microscope is typically used to detect a single fluorescent molecule? So it is TIRF microscope. TIRF microscope is total internal reflection fluorescence microscopy. Then next one. Using an analytical ultracentrifugation sedimentation velocity run, a researcher has calculated the Swedberg units of a part of the freshly purified 12 kDa enzyme to be 1.83. So for 12 kDa enzyme, he calculated the S value or Swedberg units as 1.83, corresponding to its monomeric state. So this value corresponds to the monomeric form of the enzyme. However, during an identical run of the remaining protein after 2 days. So after 2 days, the researcher again took his sample and did the ultracentrifugation experiment. The researcher finds that the S value increased to approximately 2.57. So from 183, it increased to 2.57, means around 2 times it has increased. What would be the correct conclusion about the enzyme in question? The enzyme is aggregated and forms a homo-octamer. It cannot be because you got only 2 to 3 increase. From 1.83 to 2.57 is only twice that of or thrice that of 1.83. So it cannot be an octamer. The enzyme is monomeric but is now associated with buffer components. That is also not correct. The enzyme has undergone monomer to a homodimer transition. That could be possible. The enzyme has degraded due to autocatalytic activity. If it has degraded, then its size would not have increased. Then next one. A researcher measures the height of 50 teak trees in wet and dry habitats. Which one of the following option is an appropriate statistical test to determine if heights significantly differ in wet and dry habitat? It is student G test. Answer is option 1. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.